They will be looking at the pigment producing bacteria. So there are four of them that we need to know. The first one is the Actinomyces israeli. So Actinomyces israeli is a gram-positive bacteria that is filamentous. So it has a filamentous shape. So it's not like cocci, it's not like a rod, but like filament. And um, they produce sulfur granules.
Oh,
And the
yeah,
color,
you're recording.
yes, I am. Yes, I am.
Okay.
And the color of sulfur is yellow. So whenever you see a question talking about an organism that produces yellow-like pigment or granules, they might say yellow, might say sulfur, any one of them. That is Actinomyces israeli. And the presence of this filament gives it out. So only two filamentous bacteria exist: Actinomyces and Nocardia. Okay, and Actinomyces produces yellow granules. Staph aureus produces yellow too, but it's golden yellow pigment, not granules, pigment. Staph aureus produces golden yellow pigment. Pseudomonas is blue-green pigment. And that color is impacted by these two pigment that is secret, pyocyanin and pyovadin. So we need to know that pyocyanin, pyovadin are from Pseudomonas aeruginosa, and they bring about the blue-green pigmentation of Pseudomonas. The last one is Serratia mercescens. So Serratia mercescens produces red pigment. Serratia, red pigment. So going over it again, Actinomyces israeli, yellow like sulfur granules. Staph aureus, golden yellow pigment. Serratia mercescens, red pigment. Pseudomonas, um, the blue-green pigment due to release of pyocyanin and pyovadin. Okay. Then we've already talked about this, so we are just going to dilate on it a little. Um, the bacteria that forms the biofilm in vivo, they include Staphylococcus epidermidis, the Viridans streptococcus, which include Streptococcus mutans and sanguinis. So sanguinis and Streptococcus mutans are examples of Viridans streptococcus that can form biofilm. We have Pseudomonas aeruginosa and the non-typable or the unencapsulated Haemophilus influenza. So the staph epidermidis, we talked about this yesterday. They can form biofilms within the catheter, intravenous catheters as well as prostatic devices. So if you have a prostatic device in the heart, commonly, so you can have biofilms in that area. If you have someone that has a hip prosthesis, you can have staph epidermidis growing on those prosthetic devices. So, but the most common one that we see are, you know, growth within cardiac prosthetic devices. So you watch out for that. Viridian streptococcus. And one thing about this staph epidermidis is that it's one of the major contaminants for blood culture because we take blood culture from injection through the skin. So, and it's one of those normal flora within the skin, so it can get into the blood culture that we are obtaining and contaminate it. So, Viridian streptococcus includes Streptococcus mutans, Streptococcus mitis, Streptococcus sanguinis. So, all of them can cause dental plaques, so they can cause dental caries. And from there, if someone undergoes maybe a dental procedure, maybe scaling and polishing, you can have introduction of those microbes into the heart, into the bloodstream, and from that bloodstream they can find their way to the heart to cause infective endocarditis, especially if that person has an underlying heart valve, problem. Maybe the person has a prostatic heart valve, or there is a damage to the heart valve already. Maybe there's a regurgitation, there's stenosis, and the person has this dental plaque, goes for a dental procedure, you can have dislodgement of those bacteria from the dental plaques into the bloodstream, and from the bloodstream they can contaminate and deposit within that diseased cardiac valve and cause infective endocarditis. So the virulent streptococcus are notorious for causing subacute form of infective endocarditis because it's not very explosive as the acute one, and it's kind of insidious in its onset, and you need like a background, you know, um, fertile site for the damage to occur. And what is this fertile site? And a previously damaged artwork. That's the commonest, presentation. So someone that has a dental plaque that's undergoing a dental procedure, and the person has an history of, any valvular condition, you want to give that person penicillin. But if the person has a normal healthy heart valve and the person is going for dental procedure, you might not need to give penicillin in that kind of individual. So the penicillin you are giving is more like a prophylaxis. So don't forget they can form biofilms within dental plaques as well as heart valves, causing infective endocarditis. So the sanguinis are notorious for causing this infective endocarditis because they can form aggregates. So the normal aggregate that you have within the valves includes the position of the microbe itself, particularly what we are looking at now, the Streptococcus sanguinis. Fibrin surrounds it, platelets come around. Then these sanguinis organisms have ability to release dextran, so they can break down some polysaccharides and they can form dextran from there. So the dextran further helps to aggregate those inflammatory components and make it attached to the valve so that they can prolong the duration of damage. That property is not shared with the mutants or the muties. They don't have that dextran, property. They don't form dextran. So that dextran helps to further merge all those fibrin, platelets, and everything together so that they can stay and fix within the valve and cause more damage. So that's all about sanguinis. You need to take note of that point based on the fact that it can, form dextran. Pseudomonas are,
Sorry, I didn't get that sanguine very well. You said it forms something about the cardiac valve or something.
so it produces dextran. So dextran can cause aggregation of those agent that you have in infective endocarditis. What is making up the vegetation is fibrin, platelet, as well as the organisms. So you have aggregation of the organisms, fibrin, and platelet together. When you are now adding dextran to that, it's like we are fixing it to the valve so that it doesn't go, so that you can have a prolonged duration of exposure of those inflammatory exudates on the valve, and they can prolong the damage that it causes. Do you get it? So that one occurs and it also produces a fertile site for the other organism to now come in, like these mutants and metis to join it within that area. So that's the function of the dextran. Do you get that, Dr. Lushola?
Yes.
So Pseudomonas aeruginosa can also cause biofilm within the respiratory tree, especially in people with cystic fibrosis or people with ventilator, shared pneumonia. So if someone has been on the mechanical ventilator for a long time, you can have inhalation of these pseudomonas and they can form biofilms within the lungs. Then contact lens too, you can form biofilms within that contact lens, and from there they can affect the cornea, causing inflammation of the cornea, breaking about keratitis. So you want to watch out for these three in people with pseudomonas aeruginosa. Then the last one is the non-typeable unencapsulated Haemophilus influenzae. So we said it that you don't typically have the typeable ones or the encapsulated ones causing majority of the infection again, because we can now develop vaccines against them, so they are more preventable. But the non-typeable one, we don't have vaccine against them yet because they don't have capsule. So they are the ones that are majorly responsible for the Haemophilus infection now. An example of that, otitis media, and they can form biofilms within the hair cavity, middle ear cavity, leading to development of the otitis media. Okay, so um, the spore forming bacteria. So we already mentioned this, the B's and the C's. Those are the spore forming bacteria. They form spore in response to harsh environmental condition. Maybe there's no water, there's too much heat, and they need to reduce their metabolism. So they just assume this spore form and they just settle down wherever they are, maybe in the soil, and they are metabolically inactive. We said the spore contains keratin-like coat, which has some constituents including dipicolonic acid that helps you um, have a resistance against heat. We also talked about the fact that it contains DNA so that the microbes can regenerate from there, and as well as peptidoglycan cell wall, because this spore forming attribute is native to the gram positive organisms. So that's that about the spore, they helps to mediate resistance against heat, against chemical, against dehydration, to prevent desiccation. And the way you can take care of any material that is contaminated with this spore, maybe we have our utensils, we have our surgical equipment contaminated with this spore, and you want to disinfect that, you want to remove all those things. The way you can do it is to autoclave at 121 degrees Celsius for 15 minutes. That's autoclaving. So you heat within this autoclave for 15 minutes at this temperature. So at this temperature, it is sporicidal. It kills off all these spores when you heat at this temperature for this time, 15 minutes. Other agent that you can use include hydrogen peroxide as well as iodine-based preparation. So you can use iodine-based preparation as well as hydrogen peroxide to remove all these spores from especially our surgical equipment. They are sporicidal. So what are these agents that are sporicidal? That are spore producing rather? The B is the C. So the bacillus anthracis causing anthrax, the bacillus cereus causing food poisoning, Clostridium botulinum, causing Clostridium botulinum rather causing botulinism and botulism, Clostridium difficile causing pseudomembranous colitis, Clostridium paphryngeus causing gas gangrene, and Clostridium tetani causing tetanus. Yeah, so most of
Hi,
these.
Dr. Omade.
Yes,
Are
Dr.
these also the organisms that, because of the spore, are able to cause infection very rapidly?
Yeah, so when they are within the environment might not be conducive for them, so they exist as spore. So you can take them in through this
Yes.
spore. So majority of the time, you actually take them into the body in this spore form. So
Uh huh.
when they get to the body, the condition of the body is more favorable. You have nutrients they can feed on and all those things. So they can now go back to the normal vegetative form and cause infection. So if somebody, is playing now and had a
Uh
cut,
huh. Uh huh.
so the cut is exposed to sand. Within the soil, you have the spores of Clostridium tetani there. So once the spore enter that wound, it has a tissue to feed on, environment, there's blood everywhere. So it can grow back from the spore form to the vegetative form of this Clostridium tetani and begin to cause the infection. Same thing for Clostridium botulinum. So sometimes you can find them as spore form within food substances or even canned food. So particularly food substances, not canned food, because what you have in the canned food is the preformed toxin. We are going to get there. So food substances, maybe like milk, honey, or anything, they can be there. So when you take them in, it changes its form and start causing the infection. Bacillus anthracis also, they inhale the spore. So once someone inhales Bacillus anthracis spore, it changes to vegetative form and start causing infection. But in this spore form, they can't cause infection. They are dormant. They don't do anything because they are trying to survive. But once they enter the body, they change. Do you get it, Dr. Francois?
Yes, yes. When you mentioned the preformed toxin, then I realized that that is what I was probably making reference to, because
Yeah.
I think I've heard of that. But then the Bacillus cereus over here, because okay, maybe I just remember something about Bacillus cereus.
Yeah. So Bacillus cereus too, you can take in the spore or you know, okay, let's say the food is contaminated with the spore. That's why this
Uh
Bacillus
huh.
cereus one is not like what's it called, like staph. Most of the time, Staphylococcus aureus one is more like very immediate. Once you take in the because what you are taking in for Staph aureus is actually the enterotoxin. So the enterotoxin is already in the food. So
So that's
once
the preformed toxin.
exactly, that's the preformed toxin. So you take it in. But this
Okay.
bacillus, it has to change,
The
from the spore,
spore.
it change, then start
Mm-hmm.
producing the toxin. Do you get it? It start
Mm.
producing the toxin.
Yeah.
So all of them have to change first before they start producing their toxins. But
Okay.
anyone that is, you are taking the toxin initially, it is going to be very rapid. Do you get it? It's going to be
Okay.
very rapid.
Okay.
I think
Yeah.
I've answered your question now. Yeah, I get what
You
you're
have.
saying.
Yeah. You have. Thanks.
Okay, so the virulence factor, yeah. So I was able to check the meaning of virulence. This is there's even they have even stated it here, Dr. Francois. So when I was reading I didn't really pay attention to this statement, but it's very important. That was why when you asked me that question, I was able to, because when I looked at the function of all these things, I noticed that they are not particularly causing the infection. So we can't say evidence is
Mm-hmm.
good. It's just help them
Yeah.
survive. So it's just things
So
that
they
help
are
them survive.
protective for the organism,
Exactly.
for the bacteria. Yeah.
Exactly. So they are protective for the bacteria. That's why they make this statement. They promote
Yeah.
evasion.
Evasion of host immune response.
Exactly.
Mm-hmm.
So, but anything that actually makes it cause that damage, we tend to say that one is, a pathogenicity, like a pathogenic factor. So
Yeah.
yeah,
I
two
get it.
different things. Yeah. So four different virulent factors: the capsular polysaccharide, the protein A, the IgA proteins, and the M protein. So these guys are not present in one organism. Different organisms, share these things. But the most important one is actually capsular polysaccharide. So you guys can see capsular polysaccharide question in your exam. You can see. Yeah. So the capsule is made up of polysaccharide and it is highly charged, hydrophilic structure. So that's that. It majorly acts as barrier against phagocytosis and complement lysis. Yeah. So we explained why it can protect against phagocytosis because they don't contain protein. It is polysaccharide. It is carbohydrate. And macrophages don't engulf carbohydrate directly. You have to opsonize them before they can be engulfed. It also protects against complement-mediated lysis. By the time we are doing immunology, we are going to talk more about membrane attack complex. So membrane attack complex is just like aggregate of some special complement, particularly C5 to C9. They come together and they make pores within the cell membrane. But something that has a capsule, it is very difficult to make pulse through your capsule because your capsule is not a cell membrane. Your capsule is there's an outer structure that is even protecting the inner cell membrane. So because of that capsule, you cannot make pulse through the complement. So you cannot achieve complement mediated lysis. And it's a major determinant of virulence. So an organism that has this capsular, covering will be very, very virulent. So it's very, very difficult to control that kind of organism. Yeah, so the next one is protein A. Protein A is very important too. They ask it a lot. Protein A is commonly expressed by Staphylococcus aureus. So we have to know that first. Whenever you see protein A, Staphylococcus aureus comes to your mind. And if you guys remember yesterday, we were talking about opsonization. We talked about that the fact that a typical antibody looking at IgG has two regions. It has the FAB region and the FC region. So the FAB region is where the antigen to the antibody is binding to, while the FC region is where the um the the phagocyte is going to bind to. So complement can bind to FC region. Macrophages like phagocyte generally can bind to the FC region also. So because of the expression of protein A, we cannot achieve that opsonization and phagocytosis. Why? Because protein A will come and bind to the FC region of the IgG, where our complement or our phagocyte is supposed to bind to. So in the process, you cannot achieve opsonization. And if you don't achieve opsonization, you cannot mediate phagocytosis again because there's no place for the phagocyte to bind to again. So that's how Staph aureus protects itself, by expressing protein A that binds to the FC region of the immunoglobulin G so that the phagocyte cannot bind to it and cause opsonization and phagocytosis. The next one is
What
IgA.
binds to FAB again?
FAB, the antigen. So um um IgA protease, that is common to three organisms. The Shin, we've talked about the Shin a lot. We've talked about them a lot, and this is not even the last time we are going to mention them. We're also going to mention them again when we're talking about the bacterial genetics. So we have to know these guys a lot. The Shin, we have to master the Shin. They also express IgA proteins. Yeah. So one thing about the IgA is that IgA is the most commonly produced immunoglobulin, although it has a very little concentration in serum because it is a mucosal antibody. You find it colonizing the mucosal tract, particularly the gastrointestinal tract as well as the respiratory tract. So that is respiratory mucosa, gastrointestinal mucosa. You have your IgA antibody lining there. Yeah. So um so that means that any organism that typically affect the mucosal of the airway or the gastrointestinal tract needs to take out IgA before they can exert their effect. So that's why strep pneumo, Haemophilus influenzae type B, and Neisseria that typically causes respiratory tract problems develop that IgA protein, so that when they want to colonize the airway, they just release their protease enzyme that cleaves off the IgA from there, so that they can colonize the respiratory mucous membrane and cause problems. So from there you can have pneumonia. You can have pneumonia from H influenza as well as Neisseria. So Neisseria tends to colonize, although it doesn't cause pneumonia, but it colonizes the airway, and from the airway it can disseminate into the brain. It can get through um, the bloodstream and cause meningococciemia. So someone that has Neisseria meningitis, that's why you tend to discover that Neisseria meningitis, for example, is is something that is transmitted through respiratory droplet, because once you inhale it, it stays within the airway, and from there it can migrate through the cribriform plate, the ethmoid sinuses to the brain and cause meningitis. So this Neisseria we are even looking at here is actually more of Neisseria meningitis. That is it. So they can colonize the airway because they have this IgA protein that cleaves off the IgA, that lines the mucosa of the airway. Then H influenza type B can cause pneumonia, by cleaving of this, as well as pneumonia. So we should not forget that IgA proteins, airway, and their infection. Then the last one is M protein. M protein. M protein is a protein that's typically expressed by the group A streptococcal organism. So the group A beta hemolytic streptococcal organism expresses M protein. Whenever you see M protein, one thing should come to your mind, and that is acute rheumatic fever. Acute rheumatic fever should come to your mind. M protein. Once you see M protein, acute rheumatic fever, M protein, myosin protein. So they express this M protein so that they can evade phagocytosis, number one. They also express this M protein so that you can develop once they express this M protein to prevent phagocytosis, we have antibodies developed against this M protein. Now, because this M protein exhibits molecular mimicry, it's very similar to the proteins within the myosin, that the myosin of the cardiac muscle, that myosin protein within the myosin is one of the filaments. So the myosin filament of the cardiac muscle is a protein. So it's very, very similar in terms of their structural homology to M protein expressed by the group A streptococcal. So because following group A streptococcal infection, you already develop antibody against the M protein. So that antibodies, they can cross-react with the myosin within the cardiac muscle and cause heart damage, because they damage that myosin, thinking it's M protein, and you can have carditis developed from that. So it underlies the autoimmune response that we see in acute rheumatic fever, typically the cardiac damage that they exhibit. So, but it also helps to prevent against phagocytosis. That is the main reason why the group A streptococcus is releasing it, is to evade, protect itself from phagocytosis. It prevents the phagocyte from engulfing it. But the antibody response to that M protein causes the carditis of acute rheumatic fever. So that is that. So we talk about bacterial genetics now. So bacterial genetics are just mechanism by ways um, the bacteria can transmit genetic information from one region to another, maybe from the environment to another bacteria, or from one bacteria to another bacteria. So the first one we'll be looking at is transformation. So transformation just means a bacteria picking up genetic information or genetic material from the environment into itself. That is it. That is all about transformation. It's very easy. You don't have any complex stuff about transformation. It just means that the bacteria is picking up genetic information from the environment into itself, and it is expressing the protein. So you can have this naked DNA lying around within the environment, maybe following death or lysis of the bacteria that contains it originally. So when there's damage to that bacteria and it dies off, it can release it. Naked DNA, if it's not yet degraded, another microbe or bacteria can pick it up and it can incorporate it into its own genome and they can express it. That is the concept of transformation. And the typical organism that does transformation, that have the ability to perform the process of transformation, includes the shin. So the strep pneumo, the H. influenza type B, as well as Neisseria. Those are the organisms that are notorious for doing transformation. So if you add deoxyribonuclease to that kind of cell, it can degrade the naked DNA, preventing transformation. So deoxyribonuclease can help degrade the naked DNA, preventing transformation. The next one is conjugation. So for conjugation, you need to have the pillars. So the pillars needs to be involved for conjugation. And one thing about the pillars is that the pillars gene is encoded within the plasmid. So within a bacterial cell, we have two form of DNA. We have the plasmid DNA as well as the chromosomal DNA. So the plasmid DNA is not a chromosome; it is an extra chromosomal DNA. So looking at this cell now, the one we have on top, the one that is looking like purple, is the chromosomal bacterial DNA, while the one that is beneath it is the plasmid. So any bacteria that has a plasmid can form a sex pillars. The sex pillars is responsible for forming what we call a mating or a conjugal bridge. So that
Dr.
bridge,
Babadi, sorry, please. I'm a bit lost from the beginning.
Me
Sorry.
okay,
too. Me too. Me too.
okay. So you guys got the statement that I made about bacteria containing two DNA. So I said they have the plasmid as well as the chromosomal DNA. Do you guys get that? Yeah.
Okay. All right. Yes.
So the plasmid is not a chromosomal DNA. It is not a chromosome. So
Okay.
we call it extra chromosomal DNA. So now the plasmid contains so many genes, but one of them is the gene for the sex pilus. The pilus, the pili that we talked about when we were talking about the appendages. You remember? So that the gene that codes for that pilus protein is encoded within the plasmid. So that means that any bacteria that has a plasmid can form the sex pilus. That pilus, whenever it is expressed, allows for formation of a mating bridge. You can see this picture. You can see this bridge in between. Hmm? Can you see this bridge in between them?
Yeah.
So it is due to the fact that they have the pilus. So it is pilus that helps to form that mating bridge. Now,
Mm-hmm.
we have different forms of bacteria. We have three different forms of bacteria. We have the F plus. So if we say that the bacteria is F plus, that means that it contains the sex pilus, and it means that it has a plasmid. That's what it means. If it doesn't have the plasmid, it cannot form the sex pilus, and it cannot, undergo um, formation of the conjugal or the mating bridge. So once a bacteria contains a plasmid, we call it the F positive, cell, or it has the F positive configuration. So is that clear? Do we understand that?
Yes.
So
So you said
Yeah.
that bacteria F positive indicates that you have the plasmid, but you also said the plasmid has the gene for the sex pilus.
yes, so we are still saying the same thing. So if you have a plasmid, that means
Okay.
that because the plasmid codes for the sex pilus. Do you get it? So that means that you'll be able to express that sex pilus and be forming the mating bridge. Do you understand?
Okay.
Yeah.
Okay.
So
And then these two circular, are those the two DNA that you were referring to?
yeah, so the one on top,
The
those ones on
uniform.
top, those ones on top are the
Mm-hmm.
chromosomal DNA. Those ones on top are the
Okay.
chromosomal DNA. You can see the one beneath. That's the plasmid. Mm.
Okay.
You can see that the one that is containing the plasmid is the F positive one. You can see.
Yeah.
So that makes sense because it has a plasmid that can code for the sex pilus. That's why it's F
Sex
positive.
pilus. All right. Okay.
So the one that doesn't have the plasmid cannot code for the sex pilus, so we call it F negative. You can see. So now conjugation wants to happen between these guys. Now, yeah, so as I was saying, I said three forms. So you can have the F negative form, F positive form, as well as the HFR form. So this HFR form is called the high frequency recombination form. So what that means is that you've had um, um, integration of this plasmid into this bacterial chromosome, into this chromosomal DNA. If you once you integrate or you combine your plasmid with this chromosomal DNA, we are going to have formation of the high frequency, recombination form, which is the HFR. Now look at this. I want you guys to look at what is below. So look at what is below here. You can see this HFR form. Can you guys see this? Can you guys see this?
Mm-hmm.
So it's like containing the mixture of the plasmid. You can see this original plasmid. Look at this plasmid, this second one. You can see
Mm-hmm.
this plasmid here. You can see that what you are having in this HFR is like you are mixing the plasmid with the chromosomal DNA.
Chromosomal DNA, yes.
Exactly. So once you integrate the plasmid into the chromosomal DNA, that is what we call the HFR, cell. That's high frequency recombination product. Yeah.
Okay.
So let's go back to the first image now.
Okay.
So you can have conjugation between a cell that is F positive and a cell that is F negative. Yeah. So you cannot have conjugation between F positive and F positive because there's nothing to transfer. Both of them have the plasmid. Why would they be transferring plasmid again? Because what you want to achieve with your conjugation is either transmission of DNA in any form, maybe your plasmid or any other one. We are going to talk about the other one, but for the F positive and F negative, the only difference between them is that one has a plasmid and the other one doesn't have a plasmid. I think that's clear, right? F positive, F negative. One has a plasmid, the other one doesn't have a plasmid, right? Do you guys get that?
Yeah, we do.
Yeah, so this F positive one, because of the fact that it has a plasmid, it produces the sex pilus. The sex pilus forms a mating bridge, and you guys can appreciate the mating bridge here. So the mating bridge allows transmission of this plasmid into the other cell. You guys can see and make that other cell F positive. That is what you want to achieve. Note that in that process, you are not transmitting the chromosomal DNA. You are not. You are only transmitting the sex pilus, the plasmid rather, the plasmid. So making the other cell that is F negative, F positive. Can you guys see it? Can you guys see the image?
Yeah.
So that is what we call the
Let's see.
exactly. So that is what we call conjugation between F plus and the F negative. That's what happens. There's no transfer of chromosomal DNA. Now, you can have transmission between the high frequency recombination form and the form that is F negative. Yeah. So, what is happening here? We have a high frequency recombination form, that is a bacteria that has incorporated its own plasmid into the bacterial DNA. Now, what that bacteria is actually trying to do is to transmit a portion of its own chromosomal DNA to the F negative form. Now, you guys see that the one we're talking about for F plus and F negative is that F plus is transmitting plasmid, is not transmitting bacterial DNA, by bacterial chromosomal DNA. How do you transmit that chromosomal DNA? Not plasmid. How can you transmit bacterial chromosomal DNA? The only way you can transmit chromosomal DNA is to incorporate your plasmid into the chromosomal DNA, because plasmid is something that helps you move, the DNA outside. Your plasmid is what helps you move your DNA outside. So the only way you can move out your chromosomal DNA is that incorporate your plasmid into that chromosomal DNA so that it can form the high recombination form, the high frequency recombination form. So what now happens is when a cell that has already done that, you know, comes together with F negative cell, what will now happen is a portion of the plasmid is going to lead the way. In the process, it's going to drag along portions of the bacterial chromosome DNA that it wants to transfer to the other cell. In the process, it's not going to, transmit all the portion of the plasmid. It's only a leading portion that, comes out. The other portion that will follow that plasmid segment is the chromosomal DNA. So at the end of the day, it's going to introduce the chromosomal DNA into the F negative cell, but it will not bring the plasmid into it. So you are going to form a recombinant F negative cell that only contains the chromosomal DNA but not the plasmid. So it will be recombinant, but it will be F negative. Please, I hope I've not confused you guys.
Not really, but it's not super clear. So I think I get the first part, the F positive and then the F negative. So
Okay.
now let me see if I get what you are saying about the high frequency recombination cell, right? So this is between, it also starts initially between the F positive and then the F negative, right?
Which one are you saying? Yeah,
The
exactly.
high frequency
Yeah,
recombination.
yeah.
Okay, so now that I'm looking at it, my first question is, why is it that over here the plasmid has different colors or whatever as opposed to the one on top? Is it the same thing? Are they
Yeah,
both just plasmids and
they are
they
both
just use
plasmid.
different colors?
They are both plasmid. They are both plasmid.
Okay,
Yeah.
so initially for the high frequency recombination, what happens is that the plasmid is incorporated into the chromosomal DNA,
Mm-hmm. Mm-hmm.
right? So that's
Yes.
what gives you the high frequency recombination cell. And
Exactly.
then now, by virtue of the sex pilus, which is still which is still present within the plasmid, which has now been incorporated
Mm-hmm.
into the chromosomal DNA, you have the mating bridge formed, and then
Exactly.
there's a transfer of genetic material
Yes.
to the F negative cell,
Okay. But the catch
and then
there.
you.
Okay, I'm
Mhm.
listening. I'm listening to you.
No,
I'm
no, no,
listening.
no. You can say it because
Yeah.
I'm listening.
So the catch there is that it is only the leading portion of the plasmid that comes out. It's not the entire plasmid. So it's the leading portion of the plasmid that comes out, but it's going to drag along some segment of the chromosomal DNA with it. Now that leading portion of the plasmid, as well as the chromosomal DNA that it has, dragged along, all of them will now incorporate into the bacterial chromosome of the F negative bacterial cell. But because it's only a leading strand of the plasmid that comes out, not the entire side, you are not going to form a F positive recombinant cell. It's just going to contain a copy of the plasmid. It's not going to have an entire plasmid on its own like you have in F positive.
Okay.
Look
Okay,
at this.
Baba, please take that last part again. I didn't get that last part that you just said now.
Okay, now let us look at it. We've said that a high frequency recombination cell is a cell that has incorporated its plasmid into the bacterial chromosome, right? Into the chromosome. So let's just say chromosome and plasmid so that it will be very easy. You guys know that plasmid is not a chromosome, right?
Mm-hmm.
Both of them
Mm-hmm.
contains DNA, but plasmid is not a chromosome. Now, to form the high frequency recombination, plasmid incorporate into the chromosome in that cell. So once the plasmid incorporates, that cell becomes HFR cell immediately. Now, why the cell is doing that is to able to transmit part of its own chromosomal DNA to the other cell, which is F negative. Now, what will now happen? Because of the fact that it has incorporated the plasmid into that chromosomal DNA, the plasmid portion will be able to synthesize the mating bridge. So they have a mating bridge established. Now, what will now happen is that the plasmid will now bring out a section of itself, not the entire section, because it has already recombined. It has combined with it has integrated with the chromosomal DNA. So it's only the leading portion that will come out. The remaining portion will still stay within that HFR cell. Are you following now? Are you guys following? The
Mm-hmm.
remaining portion will
Yeah.
still stay. Now, you guys remember that it has combined, there's a recombination. So it has mixed
Mm-hmm.
with the chromosomal DNA. So as the leading portion of the plasmid is coming out, the other regions will be flanked by the chromosomal DNA. So it will bring about a section of that, it will bring out a section of that chromosomal DNA that it wants to bring out. Then, with the leading part of the plasmid, the chromosomal DNA, both of them will now incorporate into the chromosomal DNA of the recipient cell. Do you guys get it?
Okay.
So you are going to form a
Yeah.
recombinant recipient cell too, because it will contain the leading part of the plasmid and it will contain the chromosome of the donating cell, as well as its own DNA chromosome too. So three things within that new cell.
Okay.
Is it not still clear? Do you guys get it
I
now?
think I think it's clear. It's like whatever has happened with the F positive is now going to be transferred to the F negative. But it's just that is the plasmid, is the sorry, is the head of plasmid, what do you call it
The
now?
leading
Is the
part.
leading part of the plasmid, the
Okay,
remaining part of the chromosome, which is a flanking chromosomal gene, and like the
exactly,
bacteria itself that is not going
exactly.
into the
You know that we've said that recipient cell is F negative. That means that it doesn't contain plasmid itself. You get it?
yeah.
So what it contains only is chromosomal DNA, but it is receiving two things from the donor. It's receiving the leading part of the donor's plasmid as well as a segment of the chromosome of the donor cell. You get it? Now, that that chromosome as well as the leading part of the plasmid will now incorporate into the chromosome of the recipient. You get it? So you have a recombinant, you have a recombination because you are having combination of plasmid and chromosomal DNA. That's what you call recombinant. Do you understand it now? But it is not going to make it F positive. Why? Because you are only moving the leading part of the plasmid, not the entire plasmid. Do you get it? So it will not make it F positive. So you just have a recombinant F negative cell. Do you
Agboye,
eh?
Agboye, I understand. I
You
don't know
understand?
what other
So
people
I want you
were
guys to really
discussing.
understand it. You guys should not just tell me you understand so that we can move. If you don't understand it, let me say it again. Dr. Adeola, do you get it? Dr.
So okay, so now my question. So this recombinant F cell, F negative cell, like um, okay, actually no. So what am I even? So what is the? Okay, I
Significance.
understand the process. So exactly, like, so
Yeah,
why does it do this at one point and do this at the other point?
exactly. So the reason why you are doing this, the later one we are talking, this recombination is that you want to transmit the chromosomal DNA to a new cell. That's the reason why you are doing this. But the first one we talked about will only help you transmit the plasmid into a new cell. You get it? Now, Dr. Francois, you now appreciate the significance now. Now imagine this recombinant F negative cell wants to transmit the portion of its own, DNA, this chromosomal DNA to another cell. How do you think it will do it? It will not be able to do it because it doesn't have a plasmid, because it's not F positive. So the only way it can achieve it is to mix with another F positive cell. So it's now going to go back, come here, mix with this F positive cell. It will be existing as this F negative cell you are seeing upwards here. Can you see my cursor?
Yeah.
So, so if it mate with another F positive cell, that F positive cell will be able to borrow it some plasmid. Do you get it? So when it has that plasmid, it can now incorporate that plasmid into its own, genome, so that it can bring out a section of its own chromosomal DNA to another cell. So the take home that I want you guys to know is that when a F positive and a F negative are mixing, the F negative is going to become F positive. That is going to contain plasmid so that it can transfer to another cell. A cell without plasmid cannot transfer to another cell. That is, once you are F positive, you cannot transfer to another cell. Be it you have recombinant form or you
You
have
mean
recombinant.
F negative? You mean once you are F negative?
Yes, once you are F negative, you cannot transmit to another cell. Even if you are recombinant, you cannot still transmit to. Do you guys understand the word recombinant? Do you understand what it means? Recombinant.
Please
Like they
explain.
recombined it, but
Exactly.
yeah, I get
Recombinant
it.
recombinant means that it contains both the plasmid and the chromosome within, a cell. That not within a cell. It means that the chromosome as well as the plasmid have integrated into one form. That's a recombinant. Now, look at this F positive cell. You cannot call this F positive cell that we are looking at for the one above. We cannot call it a recombinant. Why? Because the plasmid has not incorporated into the chromosome. Do you guys get that?
Yeah.
If the plasmid incorporates into the chromosome, it becomes recombinant. And
Okay.
you guys can remember the reason why I said they do that recombination. They do that recombination so that they can transfer their bacterial chromosome, that chromosome, to another cell. If you don't do it, you cannot transfer your chromosome. You can only transfer plasmid. Do you get it?
Yeah. So that's
Mm.
about it. How questions surrounding this? How they be framed?
I can't really remember. I've seen one or two questions on it, but it's something you'll be able to figure out. Don't worry. By the time we start solving questions, we'll see them. We'll see them. They are very easy. Once you understand the mechanism, that's what matters. So the way they just act, they just tell you, they can tell you that something picks DNA from the outside. You already know that that's transformation. I think I've seen a question on conjugation, and I've seen a question on
Transformation.
transformation before. Yes, yes,
So there are two types of combination. Either you are doing the F positive and the F negative, or the high frequency recombination and then the F negative.
exactly. So if you do the F positive and F negative, what you are doing is just transmitting the plasmid. So once you transmit the plasmid to another cell, to the F negative cell, it becomes F positive immediately because it's able to have its own plasmid. Hmm? That is it. So once this guy has its own plasmid, it can do anything. Once it has the plasmid, it can become recombinant. Do you guys get that? It can become
Yeah.
recombinant. If it becomes recombinant, it can transmit its own chromosome. If it doesn't want to become recombinant, it can transmit the plasmid to another cell too. Do you guys get that? Very easy.
Yeah.
So, so if a cell,
If it doesn't want what, Dr. Babawale, sorry.
I can't even remember what I said. It can become recombinant and it can become, um, you know, it can incorporate its plasmid into the chromosome and can become recombinant. Do you get that? It can incorporate the plasmid into the chromosome and can become recombinant. Hello?
So that gives it the ability to be able to change other bacteria, right?
Yeah, so it can
Because
donate.
what I'm getting from all these things is that it's the ability of one bacteria to either change the morphology of another one or make it have resistance to another thing or help in its own proliferation. So that's what I get from this one. So F positive that has the pilus tries to now conjugate another F negative to be able to produce itself, like replicate itself in F negative. Right? So that F negative now becomes able to also conjugate other ones. Is that it?
Yeah, so it depends on what is happening. It depends on what is happening. If it's F positive and F negative, F positive and F negative, F positive will transmit the plasmid. So if it transmits the plasmid, you are just making it have the ability to transmit its DNA to another cell. That is it. Now,
Okay,
if
so if it's the other, what's the other scenario we can have?
the other scenario is that once it has that plasmid, it can incorporate that plasmid into its own chromosomal DNA and form recombinant form. So with that recombinant form, it can transmit the chromosome and not the plasmid.
Oh, so instead of waiting for you to have plasmid, why don't I just produce one that already has my plasmid and let's just be replicating on? Is that it?
Yes. So the thing is,
Oh,
guys,
okay.
this thing is not that complex, oh,
No, it's
because
not complex. The truth is, Dr. Francois, you asked that there are questions on it, and when you see pillows, you just think this is conjugation. Like it's not that deep that you have to overthink and think and think it. Sure. But there are questions on it. Sure.
yeah. So I want you guys to understand it, and what I'm trying to say, because I know that it's this down part that is confusing. So when a recombinant is transmitting to F negative, it doesn't make it F positive. It doesn't. It just makes that F negative another recombinant. Because we've said it that recombinant only transmits a portion of the plasmid. You need the whole plasmid to move before you can form F positive. So for this recombinant to become F positive, it needs to combine with another F positive cell that is not recombinant. Do you guys get it now?
This last statement confused me. For it to do what? Say it again, sorry.
For it to contain plasmid and become F positive, it needs to combine with F positive cell. F positive cell.
Okay, that makes sense. That
Yeah.
makes sense. So that will be recombination.
Yeah.
Okay.
So maybe when we see questions, maybe it's going to be clear. When we see questions, yeah. So the next one is transduction. So transduction is of two types, generalized and specialized. So generalized and specialized transduction. So what happens with generalized transduction? So generalized transduction is a packaging error. We have something we call the bacteriophage. So bacteriophage are just viruses that contains their genome, and that genome is surrounded by a capsid protein. That's it. So what we are seeing here is a bacteriophage. So for the generalized form of transduction, what we have is a packaging error, and it is mediated by lytic bacteriophage. What happens? Whenever that lytic bacteriophage affects a bacterial cell, because of the fact that it is lytic, from the word lytic, it destroys the bacterial cell genome, the DNA, it cleaves it. It cleaves it. So this DNA, mind you, it can be the chromosomal DNA, it can be the plasmid. You guys can see that they did not specify. Maybe it's chromosome or plasmid. It can be any one of them. Both of them contains DNA. So the lytic bacteriophage, affect the bacteria, then, cleaves off the bacterial DNA. Then the next thing is to now take over the replication, the transcription, and the translation mechanism of that bacterial DNA, of that bacterial cell. It takes off, it takes up all the actions by itself. So it's doing everything by itself. It's no longer under the control of the bacteria again. So it's going to replicate all the DNAs, replicate them, transcribe them into mRNA, and also convert them into protein by translation. Of course, there's going to be a mix, because ideally the reason why this lytic phage is doing this is to destroy this bacteria, then use the machinery of this bacteria to be able to replicate its own genome. That should be the ideal thing. But that process undergoes some error sometimes, so that when it's trying to now use the machinery of the bacteria after cleaving off its DNA to replicate its own, lytic DNA, it in the process incorporate that bacterial DNA that has been cleaved off into its own DNA. So they are packaged, with its own DNA. Guys, let me come again so that I don't confuse you guys. When the bacteriophage contaminates, or sorry, infect a bacterial cell, the first thing it does, if it's a lytic bacteriophage, it cleaves off that bacteria, cleaves off the bacterial DNA rather, cleaves off the bacterial DNA, so that the replicating machinery will only be used to replicate the DNA of the lytic bacteriophage. But you know that whenever you want to, after you have, replicated your DNA, you need to transcribe it and convert it to protein. Then you package the protein, to form a new organism. That's the way it should be. Now, in the process of um, replicating DNA, and forming the protein, it also replicate the DNA of that bacterial cell, convert it to mRNA, convert it to protein, and package the protein alongside with itself. So you are going to be having formation of prophage that contains product of the genome of the bacteriophage itself as well as the bacterial cell, more like a recombination, because it's a packaging error. Instead of you to ignore, the bacterial DNA, you mistakenly package it alongside with the DNA of the bacteriophage. You replicate everything together, you transcribe them, and you translate them into protein. And at the end of the day, they are going to form prophage. So all these things you guys are seeing that looks like microphone here are prophage that contains product of the bacterial DNA as well as the lytic DNA, because of problems with packaging. Instead of you separating out the bacterial DNA, they fail to separate it out. They incorporate it into their own DNA, and they produce a product of a mix of the bacterial DNA as well as the lytic DNA. Once that happens, immediately it kills off that bacterial cell entirely. Not only the DNA now, it lyses that cell to release the prophage. So the prophage matures because one thing about this bacteriophage, we've said it that it contains only the genome and capsid protein. That is all. It doesn't contain fat, doesn't contain carbohydrates. So once you are able to synthesize the protein, which is the capsid protein, they just form that protein surrounding the genome. You have formed a complete bacteriophage. That's all they need. They don't need to be getting fat anywhere. So once you form the prophage, the protein matures into the bacteriophage. It cleaves off the bacterial cell entirely. It gets out and infects a new bacterial cell. So in the process of infecting a new bacterial cell, it will not lyse that one. It will just incorporate that bacterial genome that has gotten from the previous one into the new one. Does this make sense to you guys?
Sort of.
Yeah.
The part I don't really get is where, so after the cleavage of the bacterial DNA, and then we have the bacterial DNA being packaged into phage capsids. This release of new phage from the live cell, like, release into where? Because the next thing I'm seeing is that there's another lytic phage attaching to another cell. The link here is broken for me. The lower part.
Yeah,
Like,
so
I'm not sure.
you are looking at another image. The one for generalize is confined to this 6 image that you are seeing here.
Yes, I'm looking at the first 6.
Exactly. So once you have the
Uh-huh.
bacterial DNA being packaged into the prophage, now the prophage they will have to break out from
The
that
cell.
bacterial cell. They have to break out from the bacterial cell so that they can infect
Mm-hmm.
new bacterial cell. You understand? So they break out from the bacterial cell to infect new bacterial cell. So when they infect new bacterial cell, because they already contain a mix, their own genome as well as the previous bacterial genome or DNA. So they just transmit that bacterial genome to the
Uh,
new cell. Do you understand?
are these phage capsids, are they like, so these little things that you were describing in the thing, is there one of them that is now attaching to this other bacteria? Is
Yes.
that what is happening?
Yeah. So look
Okay,
at okay,
okay,
let's look
okay.
at it. Let's look at it with color. I think that will make sense. Now, you look at the bacterial DNA, you can see it's blue. The
Yep.
prophage, the bacteriophage DNA is red. Mhm.
Yeah.
Now, look at the packaging. So, the bacteriophage use the replicating machinery of the bacterial cell to proliferate its own chromosome, its own DNA.
Mm-hmm.
So, in the process, it's going to
Mm-hmm.
package the bacterial DNA with its own DNA into, because
Mm-hmm.
at the end of the day, when they replicate their own DNA, they are going to form protein, right? The
Mm-hmm.
protein is what is forming the capsid because we've said it that protein, the capsid is protein. We call them capsid protein.
Mm-hmm.
And everything about the bacteriophage is just their DNA surrounded by a capsid protein. Do you understand?
All right.
So
Mm-hmm.
so when it proliferates its DNA, it's going to convert some of those DNA into protein, right? You know that. So when it converts them into protein, it's going to form capsid, right? So the capsid will now surround its own remaining DNA. You get it. But in the process of packaging those DNA into the capsid, it will mistakenly package some of the bacterial DNA into the capsid too. Do you get? Hello?
Yeah, I'm here. So initially, it was going to just take over the DNA, the bacteria, and destroy the bacteria's DNA,
Exactly.
ideally, but now
Exactly.
mistakenly it's incorporated. Okay, I get it.
So now that means you are going to have some prophage that will contain bacterial DNA in them, not bacteriophage DNA,
Viral.
because they'll be
Mm.
surrounded. All of them will be surrounded by the capsid of the bacteriophage. They'll be surrounded because bacteria don't have capsid protein. It is only the bacteriophage
Yeah.
that can form capsid protein. So you will see that some of those prophage contains DNA of the bacteria, which is blue. Some of them contains DNA of the prophage, which is red. You can see. You can see. Can you see it?
Yes, I can. Yeah, I can see.
Now, the one that contains bacterial DNA, all of them will now break out. They will break out. They will form new bacteriophage. They will form bacteriophage. But what I want you to appreciate is that some of them will form bacteriophage of this initial one. bacteriophage that contains red DNA, like this initial one that is affected. You can see. Dr. Francois, you can see this. Can you see this one?
Yes. Yes, I can.
Which will be the lytic phage. Now,
Mm-hmm.
the prophage that contains the bacterial DNA, they will differentiate into this one. That is not a lytic phage. That is not a lytic phage. Hmm? Because the
Mm-hmm.
DNA that is containing is not the DNA of the bacteriophage. It is the DNA of the bacteria that has previously affected. So
Okay.
So once that one affects a new cell, it's going to just transmit that bacterial DNA into another new cell and incorporate that bacterial gene into a new cell. Why the lytic phage is going to start the cycle all over again and is going to be lysing, bacteria once it affects them.
Okay. Okay. Thank you.
So Dr. Zulia, do you understand it?
Yeah, I think.
Dr. Abisal, Dr. Oluwatobi, do you understand it? Dr. Lushola, what about you? Okay. So guys,
Dr. Wale,
yes.
I'm with you. I'm here.
So, guys, what I would just want you guys to just appreciate is that you are forming, at the end of the day, you are forming two forms of bacteriophage. You are forming a bacteriophage that contains the original bacteriophage DNA, and you are forming a bacteriophage that contains the DNA of the bacteria. So, the bacteriophage that contains the original DNA of the bacteriophage, it will continue as a lytic bacteriophage that will keep destroying bacterial DNA. But the bacteriophage that contains the DNA of the bacterial cell will not become lytic. It will just be transferring the bacterial DNA to a new cell.
Okay, I get it.
So that
Right.
is that is that is generalized transduction, and it is due to packaging error due to infection from a lytic phage. Okay, so let us go to the specialized one. So specialized one is an excision event. Is an excision error. Is an excision error. So what happens is you are going to have a lysogenic phage, which is this guy here, affecting a bacterial cell. Once it affects that bacterial cell, it's not going to lyse that bacterial cell because it's not lytic. There are two types, lytic and lysogenic. So this one is just going to incorporate its own DNA into this bacterial cell. Now, what should not happen is that normally you should have excision because this bacteriophage actually codes for some protein that can do excision at some point. So they excise, you know, um, um, what's it called? They excite, some portion of this DNA, and they convert them into protein. They excite the portion, they convert them into protein. Now, in this lysogenic problem, in some instances, you can have mistake in which the excision is imperfect. It is not excising its own DNA only. It is excising parts of the bacterial DNA, incorporating into plasmid. Normally, it should just excise its own, DNA, replicate it, convert it to protein, and move and form a new phage that contains its own DNA. But in some situation, you look at this statement here. So the DNA, the when the phage DNA is excised, flanking bacterial gene may be excised to it. So if you try and do some more reading, about this thing, although you don't have too many articles about on it online, but if you search, you can get some articles about it. You see that this error doesn't happen, commonly. It just happens once in a while. It's not a routine thing like what you have in your lytic phase. So if in the process of trying to excise its own DNA from this bacterial DNA so that it can replicate that DNA and form new bacteriophage, it now take, because we've said it that the process is just to take over the replicating machinery of this DNA because they cannot replicate on their own. So they want to utilize that of the bacterial. That's what they are doing. So, and it doesn't want to replicate the entire genome. So it just wants to integrate into this bacterial DNA, control the machinery, and use that machinery to excise itself out and replicate that its own portion only. But in some situations, it can excise some bacterial, DNA with it. So you are going to have replication of the bacterial DNA as well as its own DNA together in a cell. You guys will see that here you are forming bacteriophages that contains both the bacterial DNA as well as the phage DNA, unlike what you have here, in which you're having them in separate cells. You're having them in separate cells because this is packaging. But here, all the cells will actually have a blend of the two. Because normally what should happen is that if this bacteriophage has infected this bacteria and has incorporated the DNA into the bacterial DNA, the next thing is, okay, I am now in control of the replicating mechanism. Let me now break out my own DNA only, replicate it and leave the bacterial DNA. That's the intention. But in some situation, that excision event may be defective. It's going to excise some portion of the bacterial DNA with it, and it's going to replicate them, and they are going to translate them into protein, which is what the capsid protein, and it's going to package them, into that genome too, because it has replicated it. You guys know that once it has replicated something, to have copies of it. So some copy will form the capsid, the other copies will be packaged into the capsid. The remaining will be packaged into the capsid. So that means that you are going to be having all the cells, once you have that mistake, that means that all the cells will actually be bearing that bacterial DNA with it. So that's why you are seeing this kind of thing, because it's not excising the bacteriophage DNA completely. It is taking some bacterial DNA with it. So you are going to have, then it will keep replicating this guy. That's one thing about the lysogenic. It's not like lytic that once you have your prophage containing either the bacterial DNA or the prophage DNA, it just lyse the cell and go out immediately. These ones will keep replicating for a while, then eventually they will get lysed. It will lyse the bacterial cell so that it can escape. But the genome is going to have its own genome, which is the bacteriophage genome, and the bacterial genome. You can see that the bacterial genome is the green color. Its own genome is the purple. So it's not going to infect a new cell with the two, both the bacterial genome and its purple genome. Can you guys you see, guys, I don't want to read all these things for you, because if we are reading all these things, you might not be able to understand it. That is why I'm trying to explain it.
Iron is where is
No,
viral
I think
coming
it's
from
because
when we
this
are
is new
talking
to me.
bacteria?
So
Yeah?
it's a little bit.
Hello?
Yeah.
What did you say, Dr.
My own
Oluwatomi?
is where is viral coming from when we are dealing with bacteria?
Ah,
Like,
like I mentioned it, Dr.
The
Oluwatomi.
the
I said,
bacteriophage is a
no, I was
virus.
yeah,
distracted. That's why I didn't stop when you answered. I'm sorry.
the bacteriophage is a virus. The bacteriophage is a virus. And you know that the single component of viruses are what? Their genome, DNA or RNA, and capsid. In fact, you can have some
Okay.
viruses that are even naked. They don't have capsid.
Mm-hmm, mm-hmm,
Yeah.
mm-hmm.
But bacteriophage is a form of virus that has the genome and the capsid. So that's all it needs to form itself, the capsid and the genome. So now, when they infect the cells, they do two things. Depending on its nature, is it lytic or is it lysogenic?
That's
So if
okay.
it's lytic, so what will it do? Once it infects that cell, it's going to lyse that bacterial DNA. It's going to, take over the replicating machinery to replicate its own bacteriophage DNA and form proteins from there. What are the proteins that is forming? The capsid protein. So after it has formed the capsid protein, the next thing is now to package its own DNA into those capsid protein. But in the process of trying to package its own DNA into those capsid protein, it will package bacterial DNA into some of those capsid protein. Do you guys get it?
That
Because
makes sense to me.
yeah, so that means you are going to be having some cells having bacteriophage DNA, some cells having bacterial DNA. So at the end of the day, they are going to burst out of the bacterial cell to mature into bacteriophage. But some bacteriophage will have bacteriophage DNA, some will have bacterial DNA. So if the one that contains bacterial DNA now affect another cell, it's going to transmit that bacterial DNA to the new cell. Now,
Mm-hmm.
Now, if the bacteriophage that contains bacteriophage DNA affect another cell, what do you guys think it will do?
If, if you have both, right?
Do the same thing that it was doing to itself before.
Exactly. It's going to do what it's doing initially again because it is a lysogenic.
Okay.
But do you get it now, Dr. Oluwatobi? So
Yeah,
it's
like
going to
the way it gave itself to that one, it also kind of like likes this one too that is
exactly,
entering,
you
and then
get
try to do the same thing.
the same
Okay.
thing, the same thing. But if it contains bacterial DNA, it's not going to do that because it's not a lysogenic phage. It's a phage,
To
but it's
just
not
the
lysogenic.
transmission.
It's just going to transmit it. Very easy. That is it. But for the lysogenic one, they transmit too. But this one is going to transmit, in addition to the bacterial DNA, is going to transmit its own phage DNA too. Because this phage is actually containing both its own DNA as well as the bacterial DNA in one cell, in one phage. Because the problem here is excision. It is when it is trying to excise itself. You know that it's not damaging this DNA. A lysogenic DNA does not damage the bacterial DNA. They will rather incorporate into the bacterial DNA to take over the replicating machinery. So by the time it incorporates, it wants to excise its own DNA outside only so that it can replicate that. But the problem of trying to excise, it will take out some of those because the error will occur. So it will take out some of those bacterial DNA with it, and it will replicate, into different cells containing a mix of both the bacterial DNA and the phage DNA. And by the time they infect a new cell, they are going to transmit both of them. You guys will see that by the time it's infecting a new cell, it is no longer a lysogenic phage. Will you guys agree with me that it's no longer a lysogenic phage? Why is it no longer a lysogenic phage?
Because part of the bacterial DNA has been incorporated
You
into it.
you get it, Dr. Francois, because part of the bacterial DNA has been incorporated into it. So it will just transmit both of them into the new cell. That is specialized for. That is specialized for. So forget about the whole story. If you read it, you will see that, okay, let's read it. So it is an excision event. Lysogenic phage infects the bacterium. The viral DNA, which is the DNA of the lysogenic, incorporates into the bacterial genome. Okay. When the phage DNA is excised, flanking bacterial genes will be excised with it. DNA is packaged into phage capsid and they can affect another bacterium. Now, this DNA is a mix of both the phage DNA as well as the bacterial DNA. Now, one thing I just want us to pick here is that this is a modality for transmission of exotoxin, for exotoxins like the group A streptococcal erythrogenic toxin, the botulinum, the cholera, diphtheria, and Shiga. So that's the code is ABCDX. 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 So A is the group A streptococcal erythrogenic toxin. We are going to talk more about this toxin now after this area. The B is the botulinum toxin. The C is the cholera, not Clostridium, cholera. The D is the diphtheria, then the S is the Shiga. So, how did they transmit this toxin? A lysogenic phage is going to come around, is now going to excise the genome. It's going to excise bacterial genome that is containing this toxin that encode this toxin. So by the time it's taking out that bacterial genome that is encoding the toxin, it can transmit to another cell. So that's the mechanism by which um erythrogenic toxin or botulinum toxin can be transmitted into another cell that doesn't produce that toxin normally. So that is that. You have to appreciate that. And one thing about this is that this um any one of them that we are looking at that is an exotoxin, the bacterial DNA that it infects is actually a plasmid, not the chromosomal DNA. It has to be plasmid. Once it is an exotoxin, it will be plasmid. Now, the one that are here, I think all of them are like exotoxin. Yeah, all of them are exotoxin. All of them are exotoxin. Yeah, they are no endotoxins. They are exotoxins. So cholera, diphtheria, Shigella, um, botulinum. So don't be confused that we are saying exotoxin for gram negative. Don't be confused. Exotoxin can be released by both gram positive and gram negative. Because that's the next question I expect you guys to ask me. That okay, we are saying these are exotoxin and they have been released by gram negative organism. Like looking at cholera is a gram negative. So looking at Shiga toxin is a gram negative. Why are they releasing exotoxin? Exotoxin can come from both gram negative and gram positive. but gram-positive only express exotoxin. They don't express endo. Endo can come from gram-negative only, but exo can come from both. Yeah. So I'm happy you guys understand that. Yeah, so let's go to the last one. The last one is transposition. So what we have talked about is specialized and generalized form of transduction. Yeah, so transposition. So transposition is jumping genes, very easy. So it just means that a section of the DNA, maybe the chromosomal DNA or plasmid, a section of that DNA copies itself, excises, and moves to another region. So it can move to another plasmid or move to a bacterial chromosome DNA. So you guys can see this transposon, this jumping gene. You guys can see this blue red one. You can see this. Can you guys see this? Hello?
Yeah.
So it copies itself and move to a section of the bacterial chromosome. You can see the target site. So by the time it fixes itself to the target site, you have this. You have this. Yeah. So that's that about transposition. Transposition is important, particularly when we are talking about antibiotic drug resistance. So when we are talking about drug resistance, is due to the fact that some genes that code for drug resistance can actually move from some segment of the plasmid into the bacterial chromosome. They can move from the plasmid to the bacterial chromosome, and they can express protein that can help with antibiotic resistance. There are so many ways by which antibiotic resistance can be mediated. So there are so many ways. So that gene is going to encode protein that can do that mechanism. Yeah. So there's so many ways. For example, we have something we call MECA gene. So some of these transposome can code for um, um, another type of penicillin binding protein, such that when the antibiotics bind to that binding protein, they don't inhibit its function. So it will still retain its transpeptidase activity. That's one mechanism. There are so many mechanisms. So yeah, so they gave us an example here. They said critical in creating plasmid with multiple drug resistance and transfer across species line, particularly TN 1546 with vancomycin from Enterococcus to Staph aureus. Yeah. So Enterococcus generally they are resistant to vancomycin. So if you want to produce Staph aureus that is vancomycin resistant, how can you do that? So transposon 1546 encodes vancomycin resistance. So once it transmits from the plasmid of Enterococcus into Staph aureus, you know, that
Oh,
Staph
so
aureus.
that Staph aureus automatically resistant to vancomycin. That's Staph aureus, right?
Yeah, exactly, exactly,
Oh, so
exactly.
before we leave this part, because I saw a question, and the question was, he cited one of these transformation and all that, and then the follow-up question was like, would he be able to transmit to their progeny? Do you
Which
get
one is
like
that?
which of these? I can't remember. God,
Say it
the
again.
way it was.
Say it
So it
again.
was like it's I think it's simulated conjugation or phenotypic assessment or one of those things. And it
Okay.
was now like, would it be able to transmit to subsequent progeny, like things that have to do with changing the entire chromosome? Would it be able to give it to subsequent bacteria that it is producing? I pray I find that question. I'll just drop it.
Yeah,
I pray
yeah.
I
It's,
find
it's
it.
likely it's likely to be part of those assortment of a team. All those influencer stuff.
Okay, so no, this bacteria now they cannot transmit it to other types of bacteria. Is that current bacteria that will have that transformation, Abi?
Yes, exactly.
Like if he has a child now, like you get what I'm saying, is it going to give that ability, like this staph aureus that we are talking about now?
Mm-hmm.
If the staph aureus produces another staph aureus that has not been infected by enterococcus but came from this one that is resistant to vanc, would it also have that ability to be resistant to vancomycin?
Yes, they can. The thing is, once that microbe contains this transposon gene, those transposon gene they have ability to jump. They can move out of the cell and go to another cell. Do you get it? They have ability to jump from one DNA segment to another DNA segment. It can be within the same cell, it can be another cell. Do you understand? Hello?
Okay. So
Yes.
basically, what I'm getting from this is the bacteria's ability to proliferate or to kind of replicate itself within organisms. That is the summary of all these things happening.
Yeah. The summary is that
Okay.
you are trying to transmit gene from one bacteria to another. You are trying to transmit gene from one bacteria to another. So you can do that by so many mechanisms. It can be transformation. So transformation is actually due to that. Okay, the donor is actually dead. So it releases its own naked DNA into the environment. So the cell that can undergo that transformation goes to that area and picks it up. And it's only some organisms that can do that. Shin are the ones that can do transformation.
Thank you so much, Dr. Babawali. It's clear now. Thank
Exactly.
you.
So transposition is just that whenever you have an organism containing that transposons are jumping gene. It means that it can move that gene, and most of those jumping gene can code for anything, but most commonly they code for genes that allows for drug resistance. So once that bacteria contains that transposon can jump from that bacteria to another bacteria and transmit that resistance to that other bacteria. They don't need to be forming mating bridge. They don't need to be infected by bacteriophage. They don't need all those ones because the transposons can jump. So they jump out into the next bacteria and they transmit it. So that is why enterococcus can transmit to Staph aureus. And the transposon that we are looking at is TN1546. And what it encodes is actually resistance against vancomycin, VANC A gene, which confers resistance against vancomycin. So once it's, located within that particular plasmid of that enterococcus, it can duplicate itself, then it can, copy, excise itself, then move outside to another bacteria to transmit that gene. So it copy, excise, then goes to that next bacteria, transmitting that gene to it. Do you guys get that? That's about the transposition. It's just jumping. You don't need anything to facilitate the jumping. They just move from one cell to another cell, transmit. So whenever they are within the same colony, they can move from one cell to another. Hope it's clear to us, Dr. Zulia. Is it clear to you?
Yeah, I got it.
Yeah. So most of the time that transposition that is actually within the plasmid is within the plasmid. So it's the plasmid that contains it. Most of the time is the plasmid. Don't forget that. Okay, so what are the features of exotoxin and endotoxin? So we've been talking about some of them. The source of exotoxin is gram-negative and gram-positive. Endotoxin is only the gram-negative. The exotoxin, they are secreted from the cell. Yes. The endotoxins are not secreted from the cell because they are part of the cell wall. The only way you can get them out is by you destroying that cell. When they slice the cell, that's when you can release them. Or you can dispatch them out as blebs. So they form blebs. They now get separated from the cell and they cause, they release them into the blood and they can cause the problem. They have to detach from, that cell, either by lysis or by formation of blebs. The exotoxin are actually secreted from the cell. They are actually produced by the cell and they are secreted. That is exotoxin. Endotoxin is part of the cell. It's not being produced. It is actually part of the cell. What does it contain? Exotoxin are polypeptide. That is why you can mount immune response against exotoxin and can form antibodies. So once you form antibodies against exotoxin, what you have is antitoxin. Now, endotoxin are not polypeptide. They contain lipid A. Lipid A is not a protein. So that means that you cannot form antibody against it. That's why there's no anti-endotoxin. There's nothing like that. You can't form antibodies against. They just trigger the immune response. The cytokines that are released will just attract the inflammatory cells and they will try to clear it away. That's all they do. Yeah, you don't have antibody formation against it. Location of the genes. Now I've mentioned it. Exotoxin are located within the plasmid of bacteriophage, not bacterial chromosome, because we said it that exotoxin are actually being transmitted through which mechanism? What's the mechanism for transmission of exotoxin?
Specialized transduction.
Exactly. So specialized transduction. You guys remember that specialized transduction requires bacteriophage. So the bacteriophage actually transmits it through the plasmid, not the bacterial chromosome. But the location of the gene for the endotoxin are within the bacterial chromosome. Don't forget that. The toxicity, the toxicity of exotoxin is very high. So you just need a very small amount of dose to trigger the response. Just need one microgram. But endotoxin, it is very, very low. You need as much as 100 micrograms to trigger the same fatal dose. So it is low compared to exotoxin that's very high. What are the clinical effects? It varies. We are going to see them down now. The clinical effect of endotoxin is just constant because endotoxin will release lipid A. Lipid A activates for three things: macrophages, complement, and tissue factor. Everything at the end of the day will bring about fever, hypotension, shock will follow, and DIC. The tissue factor is going to activate the coagulation cascade that will bring about DIC. The fever is going to be from interleukin 1. The hypotension is going to be from TNF as well as histamine. So we are going to get there. We are going to see. So the clinical effect of endotoxin is constant, regardless of the endotoxin. Regardless if it's coming from E. coli, it's coming from Kleb, everything will cause septic shock. Regardless, they have a common effect. But the effect of exotoxin of diphtheria is different from exotoxin of Shigella, is different from exotoxin of diphtheria, is different from the exotoxin of erythrogenic toxin from group A streptococcus, is different from that of botulinum. You know, botulinum doesn't cause pseudomembranous formation. It doesn't cause formation of scarlet fever. So the effect of exotoxin varies, while that of endo is constant, septic shock. The mode of action is also vary for the exotoxin. We are going to, shed more light on them. Why the mode of action is due to release of TNF, interleukin 1 and 6. That's it. So we are going to see them very well. The antigenicity, so we've said it already, because of the fact that they are polypeptide, they are protein, they can induce formation of antibodies, so you can produce high titer antibodies that we call antitoxin. So that's why tetanus, exotoxin, you can have antitoxin against it. Diphtheria, you can give antitoxin against it. The endotoxin are poorly antigenic, so you don't have antibody against it. Vaccines, so you can have toxoid, because you can have you can take a piece of that polypeptide and you can conjugate it, you can do so many things with it. You can so once you take that polypeptide and you eat it, you inactivate it, they become toxoid. So once you take exotoxins and you inactivate it, it becomes toxoid. But endotoxins, you don't form toxoid against it. They don't have vaccine. They don't form vaccine. Yeah. Heat stability. So they can be destroyed rapidly. The exotoxin can be destroyed rapidly, except E. coli is stable and the staphylococcal. All these ones are not high yield, but if you want to learn it, you can learn it. Just particularly know that E. coli is stable, or from the name, it is stable to eat. The staphylococcal enterotoxin too is stable. So that's the one that causes food poisoning. So even if you cook that food that contains the staphylococcus enterotoxin, it doesn't still mean that the person will not come down with the diarrhea, because they can withstand it. Typical disease: tetanus, botulism, diphtheria, cholera. So Neisseria meningitis can cause meningococcemia, which can bring about sepsis. So that's an example of endotoxin. So someone that has E. coli sepsis, that's endotoxin. So yeah, so now let's look at those guys one by one. So we are talking about the exotoxins now. So we said here that the mechanism of action varies. So we are looking at them one by one. So some of them can inhibit protein synthesis. And they do it by two different mechanisms. Some inhibit elongation factor. You guys remember when we were talking about protein synthesis that elongation factor is important in the elongation phase. Some can inactivate, so some can inactivate 60S ribosome by removing adenine from the ribozyme or ribosomal RNA. So let's take it one by one. So the two groups that inactivate elongation factor 2 include Corynebacterium diphtheria as well as Pseudomonas. So they commonly ask this one. Dr. Olatobi, I know you're familiar with so many questions about this.
Chorus Sam. I don't the chorus Sam. And it confuses a lot. It's so annoying.
Yeah, it confuses a lot. So you have to be master.
Seriously. Yeah.
So you just have to marry those ones that share the similar stuff together. Once you can compartmentalize them, it will be easy. So Corynebacterium and Pseudomonas, both of them inhibit elongation factor 2. How do they inhibit it? They do ADP ribosylation. So once you add ADP to elongation factor 2, it inactivates it. ADP ribosylation inactivate the elongation factor 2 and halt protein synthesis. Now, what is the toxin of Corynebacterium that does that? Diphtheria toxin. What is the toxin of Pseudomonas that does it? Exotoxin A. If you want to learn the name, you can learn it. But the most important thing is remembering the mechanism of action of those toxins. That is it. That is the most important thing. Once you see Pseudomonas infection, the next thing most of the time they will not ask you what's the name of the toxin. They will just tell you what is the mechanism of action of that toxin. So how does it do? So they both inactivate the elongation factor by ADP ribosylation. So what will now be the clinical effect? Both of them will halt protein synthesis. For Pseudomonas, it's just going to cause necrosis of that cell immediately. So that is why you tend to notice that majority of the infection of Pseudomonas is actually very severe. They cause things that are very necrotic, like malignant otitis externa. There's like chronic inflammation and necrosis of that inner hair. That I say inner outer hair due to the cell death of the epithelial cells of the hair, because Pseudomonas cause death of the cells that it affects. So it causes eventual necrosis. That's why you can see them causing malignant otitis externa. They can cause ichthyoma granulosum. You see something that looks like necrotic tissue in the skin. We are going to see it when we are looking at Pseudomonas. So it causes very chronic, disease. Yeah, because of the chronic inflammatory response to the necrosis that it brings about. Corynebacterium diphtheria, brings about protein synthesis alteration. It stops protein synthesis. This protein synthesis typically, it typically affects the pharynx. So when it affects the pharynx and bring about inhibition of protein synthesis, the mucosal of the pharynx becomes necrotic. So when they die off, there's inflammatory response to that. So the inflammatory response contains inflammatory cells coming around, fibrin coming around, from the platelets forming the fibrin clot, as well as the organisms themselves. That aggregate together to form the pseudomembranes. That is characteristics of Corynebacterium diphtheria infection. So they form the pseudomembrane because of the protein synthesis that is stopped, which brings about cell death and inflammation. The inflammatory process can spread to the nearby lymph node, particularly the submandibular, the cervical lymph node, the surrounding lymph node, which brings about the bull neck appearance. So you see the neck that is swollen because of the engorgement of the cervical lymph node. So the bull neck appearance. Protein synthesis can also be stopped within the heart. So when it becomes disseminated into the bloodstream from the pharynx, it gets to the blood, it can affect synthesis of the protein of the heart, which brings about inflammation of the heart. So when there's protein synthesis alteration, there's necrosis, there's inflammation, which brings about myocarditis. It can also disseminate and affect the neurons, causing peripheral neuropathy. Don't forget that. So once you remember diphtheria, it starts within the pharynx because it's an airway disease. They actually get it by inspiration of the when they inhale the respiratory droplet. So that's how they get it. So it starts within the pharynx, causes necrosis of the pharynx, inflammation that brings about formation of the pseudomembranes, inflammation of the lymph nodes, spreads to the heart and the nerves, causing myocarditis. So that's that about Corynebacterium. The next ones include Shigella. Shigella. Both Shigella and enterohemorrhagic E. coli, they are very similar. Enterohemorrhagic E. coli also uses the Shiga toxin. Some people call it Shiga-like toxin, but you can just anyhow Shiga toxin because it similar to the toxin that Shigella also releases. What do? They inhibit protein synthesis, but not by inactivation of elongation factor. How they do their own is to remove adenine from rRNA. rRNA is also one of the components of the 60S ribosome alongside with our mRNA. So once you remove adenine from rRNA, it will inactivate the 60S ribosome. So you cannot continue protein synthesis. So once protein synthesis stop, the proteins of the GI mucosa cannot be formed. So you have necrosis of the GI mucosa, which brings about, dysentery. So you have blood everywhere due to damage to the blood cells, the epithelial cells, and everything. The blood vessels, the epithelial cells, everything becomes necrotic, which brings about the dysentery that they experience. So the dysentery also comes because of the fact that Shigella can invade the GI mucosal cells directly and damage them, bringing about the bloody diarrhea. This sugar toxin, you know, in addition to inhibiting protein synthesis and that, it can also stimulate release of cytokine. So, in response to the chronic inflammatory response, they release cytokines, and the cytokine can explain why they present with the hemolytic uremic syndrome. So, what I just want you guys to take home here is that hemolytic uremic syndrome is not only native to enterohemorrhagic E. coli. Shigella can also cause it because the starting problem is the Shiga toxin. So once the Shiga toxin is released, it causes protein, synthesis alteration. There's chronic inflammation. There's release of cytokines, and when cytokine becomes released, it activates the platelets. You start having coagulation. So when there is coagulation within the microvessels of the body, there's thrombocytopenia. So the coagulation within the microvessels causes this microangiopathic hemolysis because the red blood cells find it very difficult to pass through those tiny vessels that have been occluded by the clot. So you have the schizocyte being formed within the smear, and the red blood cells are being broken down, leading to anemia with formation of the schizocyte. So if it affects the vessels of the kidney, you have poor perfusion to the kidneys because of affectation of the small vessels of the kidneys, the arterioles, you know the segmental arteries, and all those things, bringing about AKI. So in addition to the thrombocytopenia and the microangiopathic anemia, you have elevation of creatinine and urea. So the consideration of those findings is what we call hemolytic uremic syndrome. So the person cannot have a bloody diarrhea. So once you have bloody diarrhea with that triad, that is hemolytic uremic syndrome. And bloody diarrhea can occur in somebody with enterohemorrhagic E. coli because it can damage the GI mucosa, or people with somebody with Shigella. They also damage the GI mucosa, causing the bloody diarrhea, and they also cause um, the the triad that we have talked about. So don't forget, enterohemorrhagic E. coli does not invade the O cells, but enteroinvasive form of E. coli can invade the O cells. Shigella can also invade the O cells. That is why enteroinvasive form of E. coli causes bloody diarrhea too, or dysentery, but not because of the fact that it inhibits protein synthesis, but because it can directly invade the O cells. Do you have a question? Any question before I move from there? Any question from anybody?
How are we going to remember these things?
So,
Dr. Francois, honestly, and there was a question I saw that it said, there's a part that was, I think it's traveler's diarrhea. So the person traveled somewhere. I missed the question anyways. That's why I can't remember. So the person traveled somewhere, developed diarrhea. It went on its own. They were now like, which of the following toxins acts in the similar mechanism as to the toxin that caused diarrhea in this patient? Can you
Can you
imagine this type of
imagine?
question?
What kind of question is
And,
that?
and the funny thing is, what they were testing was like this cyclic AMP.
yeah, enterotoxigenic E. coli. That's the answer. So, enterotoxigenic E. coli causes traveler's diarrhea, although it's not the only one. Giardia can also cause it, but the most common one is enterotoxigenic E. coli. So the next thing
Dr.
is for you.
Papale, you don't
Hello?
causes diarrhea by the mechanism that enterotoxigenic causes that diarrhea. Do you get?
Yeah. So I understand what you guys are saying. See, Dr. Francois, see, the only way you can learn this thing is actually, that's one thing about micro. You have to palm it. The more you look at it, the better it sticks. I did not learn it too in one day. I was looking at it repeatedly until it, stayed. You just have to keep looking at it. But I think if you have somebody, telling you, it should actually stick better compared to if you are reading it on your own. Because when I was reading it then, I was actually trying to just gloss over it. But when I noticed that they test it a lot in question, I had to go back and calmly palm it. That is it. So it's not something you can gloss over. You really need to, you know. But I feel that with time you are going to master it. Corynebacterium and Pseudomonas, they go together. Shigella and enterohemorrhagic. I think Shigella and enterohemorrhagic should not be hard for us. Shiga toxin, right? Inactivate the 60S ribosome. Then we go to the ones that are associated with fluid secretion. The thing is, one way to look at it is look at the downstream effect. What is happening? Pharyngitis, okay, is not likely to be due to water secreting or something. So that's like, okay, protein synthesis. It's only protein synthesis that can lead that to necrosis and eventual inflammation due to that necrosis. Enterotoxigenic E. coli causes diarrhea. How can that diarrhea happen? Okay, I remember that my CMP stimulates secretion of chloride into the lumen. And once you have secretion of chloride, water follows. How do you achieve that? It's due to overactivation of the adenylate cyclase. So enterotoxigenic has two toxins, the EAT labile and the EAT stable. They work differently, although both of them cause diarrhea, but they work differently. The EAT labile one is going to stimulate adenylate cyclase. Adenylate cyclase is an enzyme that cleaves up CAMP from ATP. CAMP stimulates secretion of chloride into the lumen and water follows, causing the diarrhea. For EAT stable stimulates guanylate cyclase. Guanylate cyclase produce CGMP, not CMP. CGMP normally, to me, is kind of a molecule that actually calms something down. It kind of stabilizes. So what it does is to reduce reabsorption from the lumen into the epithelial cells. So if you reduce reabsorption of sodium chloride from the lumen into the epithelial cell, that means you have abundance of sodium chloride within the sodium chloride and water within the lumen. So that can cause diarrhea, because the way you maintain normal balance is by controlling the rate of absorption and the secretion. So if there's an imbalance between that process, diarrhea can happen. So each labile stimulates the CMP that secretes. That's an active process. That's the way to look at it. Active process, CMP. But CGMP cools down. It suppresses reabsorption from the lumen, so that majority of those electrolytes stay within the lumen. Water is stayed there with them, and they can, cause the diarrhea that, it brings about. It's stable. Yeah. The code is actually keep, keep, keep. But let's continue. So for the other ones that also increase fluid secretion, the next one is Bacillus anthracis. So Bacillus anthracis toxin is anthrax toxin. One way to look at this Bacillus anthracis is that they actually form what we call E-scars. So E-scars are just like necrotic scars within the skin, and they look edematous. So to form that edematous scar, you need to increase water absorption. How can it do that? It mimics adenylate cyclase and stimulates production of CMP that causes absorption of water into those cells, causing the edematous border of the eschar. That is that about anthrax. Vibro also increase adenylate cyclase, but how does it do it? It permanently stimulates GS components. So glycoprotein, G protein coupled receptor contains so many components. It contains GS, GI, and GQ. GS and GI control CMP. So if you stimulate the GS, that's the stimulatory component. GI is the inhibitory component. So if you stimulate the GS, you have more CGMP that is formed, which brings about secretion of more chloride, more water, causing the rice water diarrhea that people with cholera exhibit. Guys, does it make sense to you? Can you guys, appreciate all those things that we are saying? particularly this fluid secretion part.
Okay, so basically they are all increasing fluid secretion by activating, all of them are activating CAMP except the heat stable.
Okay,
But
I agree
wait,
with you.
so
I agree with
yeah.
you. I agree
So
with
the heat
you.
stable is activating guanylate cyclase, okay, by decreasing the resorption. So it's working in the opposite way to, okay, so it's opposite to that of CAMP. So whereas CAMP is promoting chloride secretion and water efflux, CGMP is decreasing resorption of sodium chloride and water.
Exactly. You have learned that. That one is gone. Dr. Francois, I'm telling you, that one is gone. You've known it. See, you might think I'm joking, but it's actually the truth. When I was reading it, I was just glossing over it. I was not really, I was just thinking they can't be asking all these things. But they actually do test them. And once you've known it now, it stays with you. Anthrax, forget about the way anthrax present. Just remember the ESCA that contains fluid, edema. So it mimics adenine cyclase to increase CMP rather, and increase water absorption. The last one is cholera, diarrhea, cholera. But how does cholera does it own? It permanently activates the GS so that the GS can activate adenine cyclase, and adenine cyclase can increase CMP and can increase the absorption of chloride. That's it. So enterotoxigenic one, the stable, the each labile. Each labile has increased adenine cyclase activity, increased CMP, increased the absorption of chloride and water. Bam. The the heat stable one, the heat stable one uses CGMP. So it stimulates guanylate cyclase, not adenylate, guanylate cyclase to increase CGMP. And we said the CGMP tries to calm down everything. So it doesn't actively secrete, it rather reduce the absorption from the lumen, bringing about the diarrhea. What's the last one? Anthrax. Anthrax mimics adenine cyclase so that you can have increased activity of formation of CMP, and water can be reabsorbed, forming the edematous um, borders of the ISCA. Then the last one is the cholera. Cholera permanently activates the GS subunit of the G protein coupled receptor. Once you activate the GS subunit permanently, you are increasing adenine cyclase activity permanently, you are increasing conversion of ATP to CAMP permanently, and there is permanent reabsorption of chloride and water, um, permanent secretion of chloride and water into the lumen, causing the diarrhea, which is known as rice water. You see, you go back to the previous one we've talked about. Talked about Corynebacterium and Pseudomonas. Those are the ones that go together. They inhibit elongation factor 2 by ADP ribosylation. So protein synthesis stop, there's death of the cells. For Corynebacterium, it starts within the pharynx, so there's death of the pharynx mucosal cells. Chronic inflammation happens in response to necrosis. So when there's inflammation, there's pharyngitis. So the inflammatory cells are what forms the pseudomembranes. The corynebacterial organisms, the fibrin clots that are formed, as well as the inflammatory white blood cells that are surrounding it. They form the pseudomembranes that brings about the pseudomembrane appearance in pharyngitis of diphtheria. They can spread to the neighboring lymph node and cause bull neck appearance from the lymphadenitis, which is usually bilateral. They can disseminate into the blood and go to the bloodstream. When they affect the heart, they typically affect the heart and the nervous system. So they damage the protein, the synthesis of the protein of the cardiac muscle. There's inflammation in response to that, bringing about myocarditis. There's inflammation within the neurons, which explains the peripheral neuropathy that they present with. Pseudomonas is very straightforward. Alters protein synthesis formation of by inhibiting activity of elongation factor 2 by ADP ribosylation, just like diphtheria. It causes host cell death, regardless of the cell, depending on where it's contaminating. Easy. The next one includes our Shiga toxin and our enterohemorrhagic E. coli. They produce Shiga toxin. What does Shiga toxin do? It inhibits protein synthesis. How? It removes adenine from ribosomal RNA. Ribosomal RNA is a component of 60S ribosome. 60S ribosome doesn't work. You cannot have that protein synthesis machinery happening. Don't forget, ribosomal RNA is also known as ribozyme that catalyzes the formation of the peptide bond. You cannot have protein synthesis. The GI mucosal cell that they typically affect, they die, they slot off. You have diarrhea from there, which is bloody because of damage to the blood vessels too from the sloughing off of the epithelial cells. There's dysentery, bloody diarrhea. In response to the sugar toxin, there's release of cytokines. These cytokines causes inflammatory response, which brings about coagulation. There's coagulation, which is widespread, typically affecting small vessels. Because of the coagulation, there's thrombocytopenia, consumption of the platelets. The coagulation obstructs the small vessels, causing microangiopathic hemolytic anemia because the red blood cells try to pass through, they squeeze themselves, they become lysed, they form schizocytes, and they reduce in number, causing the anemia. When there is reduction in perfusion to the kidneys from the anemia or from the obstruction of the renal vessels, there's AKI. The triad of AKI, thrombocytopenia, and the microangiopathic anemia with the bloody diarrhea is called hemolytic uremic syndrome, which is typical of anything that can produce the sugar toxin, particularly enterohemorrhagic E. coli or Shigella. Then we start talking about these guys again, the fluid secretion. So let's go to the last one. The ones that inhibit phagocytic ability. Now, I want you guys to be careful here because this one is caused by Bordetella pertussis. So once you see, I've seen this one in question, maybe like 40% or 37% got that question because it's very unusual. Bordetella pertussis release pertussis toxin. How does it work? It inhibits phagocytic ability. It does not cause fluid secretion. It doesn't inhibit protein synthesis, but it inhibits the ability of the phagocyte to phagocytose. How does it do it? It does it very similar to the fluid secreting ones. It also activates adenylate cyclase. Just like our E. coli, our labile E. coli will do, to activate adenylate cyclase, increasing CMP. But how does it achieve its own? It does not stimulate GS like cholera. It rather inhibit GI. So once you inhibit GI, that means that there's nothing to inhibit production of CMP, from adenylate cyclase. That means that the activity of adenylate cyclase increases because you have inhibited the inhibiting factor for adenylate cyclase. GS stimulates it. GI inhibits adenylate cyclase. So once you inhibit GI, that means that adenylate cyclase increases in activity. Then you have more CMP. Increased CMP has been shown to downregulate the chemokine receptor expression. So when there's downregulation of chemokine receptor expression, there's nothing to attract your neutrophils. Neutrophils are phagocytes too. So when there's increased CMP, there's downregulation of the chemokine receptor expression because it's a form of protein. Because don't let me go there. Let me just stop there. So when there's downregulation of this chemokine receptor, you don't have chemokines in circulation. You can't attract the neutrophils. There's little phagocytotic activity. That means that your Bordetella pertussis can be playing around and be causing the infection. That is it. It causes whooping cough, in which the child coughs on expiration and whoops on inspiration. So you hear something like, as if the child is wheezing. So whoops when he's trying to take in air, and when he's trying to expire, there is coughing. So in adults, it's known as the 100-day cough, and there's actually a post-tussive emesis. Yeah, that one is not specific to anything. Just know that Bordetella pertussis inhibit GI, increasing CMP that downregulate chemical receptor that inhibit attraction of neutrophils, which brings about reduced phagocytic ability. That is that. The last one, not even the last. The next one, the ones that inhibit release of neurotransmitter. I know that all of us know these ones. We know this one. The two that inhibit neurotransmitter release Clostridium tetani and Clostridium botulinum. Clostridium botulinum and Clostridium tetani. Clostridium tetani and Clostridium botulinum. It's different from the way Clostridium perfringens work. They are different. Different from the way Clostridium difficile work. They are different. So, the Clostridium tetani release tetanospasmin. So forget tetanolysin now. Tetanospasmin is our focus, not tetanolysin. Spasmin. Clostridium botulinum release botulinum toxin. Both of them inhibit the activity. They are protease, enzyme that can actually cleave off what we call the snare protein. So forget about the full meaning of the snare. Just know snare protein are proteins that actually mediate fusion of the neurotransmitter with the presynaptic membrane so that it can be released into the synaptic cleft to act at the neuromuscular junction. So if an individual have cleaved up the snare protein, there's no way the neurotransmitters can fuse. The vesicles of the contain the neurotransmitter can fuse with the membrane and be released into the synaptic cleft. So what does it now happen? What does it now lead to? What happens? Clostridium tetani, when it does that, it's actually specific for inhibiting the release of neurotransmitters vesicle containing GABA and glycine. These are inhibitory neurotransmitters. So it inhibits the release of this inhibitory neurotransmitter from inhibitory interneurons. So those inhibitory interneurons are actually there to regulate the activity of the alpha neurons that cause muscle contraction. So when you inhibit release of GABA and glycine from the inhibitory interneuron, that means there's no way they can inhibit outflow from the alpha neuron. That means that the activity of the alpha neuron becomes unchecked. So they have increased discharge from the alpha neuron. Increased discharge from the alpha neuron, leading to spastic paralysis, rhesus, sardonicus, trismus, and obstructionus. So obstructionus is when you have uncontrolled contraction of the extensor muscle of the back. Trismus is contraction of the masseter muscle of the jaw. That's the first sign that we call the locked jaw. So they cannot open their mouth. Rhesus, sardonicus is when you have unopposed contraction of the muscles of the face. Spastic paralysis is when you have, weakness. You cannot move that part, but it's spastic with increased tone. It's not flaccid, it is spastic. Increased contraction. Yeah. So, Clostridium botulinum also inhibit release of neurotransmitter. But what are the neurotransmitters that it inhibit? Acetylcholine. So particularly everything you are going to be having here is flaccid paralysis. That is it, not spastic. They cannot contract at all. In Clostridium tetani, they are contracting too much. But here, they are not even contracting at all. So, what are the two forms of Clostridium botulinum? The infant and the foodborne. The infant is the one that you are ingesting the spores from soil, from honey. So you have to produce the toxin following the injection in vivo. So it takes time to manifest. The foodborne is usually immediate, because you are ingesting the preformed toxin from canned food usually. So if you read the history and they are seeing the person consumed a canned food, you know that that's likely to be foodborne. But if the person takes maybe soil or maybe something that's contaminated with soil or honey, it's likely to contain the spores. So that would be the infantile form, because it's infant that tends to put different things in their mouth. So it inhibits release of acetylcholine at the synaptic site. So there's no acetylcholine to stimulate the muscle and cause muscular contraction. So they have facet paralysis with botulinum. So you use this botulinum toxin in so many situations. For example, someone that has a chalacea, you can inject it to relax, the tone of the lower esophageal sphincter, because it tends to bring about flaccid paralysis. You can use it in so many conditions, so many. Diffuse esophageal spasm can treat it with botulinum injection. So, guys, we are going to stop there. I thought we were going to move beyond this stuff, but so many
Oh,
Okay.
that's
explanations.
good. That's good.
Bye-bye.
5 pages.
Hello?
We need to go and